कर देते हैं है नहीं पता चलने वाला सब व्हाट्स मैंने लिंक में दे तेरे स्क्रीन स्टार्ट है मैडम स्क्रीन स्टार्ट ही है आपकी स्ट्रीम स्टार्ट है आपकी स्ट्रीम स्टार्ट है मैडम ऑलरेडी कब से स्टार्ट है 12 मिनट से पहले से स्टार्ट है Good morning, respected teachers and dear friends. The seminar topic assigned to me is non-culture based methods of resistance detection. My moderator is Dr. Aarti Kapil. So coming to the burden of antimicrobial resistance, antibiotic resistance is one of the major problems globally, such that it has become a pandemic. It results from the excessive consumption of antibiotics due to overprescription, self-medication, over-the-counter accessible antibiotics, absence of standardized guidelines for antibiotic usage, lack of sanitation and hygiene practices, and also the use of counterfeit drugs. 2 million people have become infected with antibiotic-resistant bacteria, resulting in at least 23,000 deaths each year. This is as per the CDC 2013 information. This slide shows the WHO global priority list of antibiotic-resistant bacteria to guide research, discovery, and development of new antibiotics. We see that in priority 1 are the critical bacteria, that is Acinetobacter baumannii, which is carbapenem resistant, Pseudomonas aeruginosa, which is carbapenem resistant, Enterobacteriaceae, carbapenem resistant, and third generation cephalosporin resistant. In priority 2, there's Enterococcus faecium, Staphylococcus aureus, Helicobacter, Campylobacter salmonella, and Neisseria gonorrhoeae. The need for rapid identification of antimicrobial resistance is it's an urgent need because traditional approaches take 24 hours for obtaining growth from clinical specimens. An additional 24 hours is used for downstream isolate characterization, that is biochemical identification and phenotypic susceptibility testing. Therapeutic decisions are made empirically in the meanwhile, and use of broad empiric treatment regimens is what drives emergence of multi-drug resistance. Phenotypic results are time-consuming. Classical antibiotic susceptibility tests, example the Kirby-Bar diffusion or the E-test, require 1 day of incubation. Liquid microbot dilution, which is the gold standard for AST, is laborious, and results are available in 6 to 24 hours. And this requires prior bacterial culture derived from solid phase incubation. On the other hand, genotypic AST or molecular characterization is rapid, and results can be obtained in 1 to 3 hours and can be used to investigate presence of a given gene or point mutation and direct support to ensure that optimal treatment or control strategy is undertaken in a timely manner. Molecular characterization also allows an indirect method to aid epidemiological investigations following an outbreak, and molecular characterization of antimicrobial resistance determinants is also used for local, national, or even global surveillance of antimicrobial resistance. So in the genotypic methods of non-culture based resistance detection, we have quantitative real-time PCR, multiplex PCR, DNA microarray, next-generation sequencing, and fluorescent in situ hybridization. Among the phenotypic methods of non-culture based resistance detection, mass spectrometric techniques may be used. So coming to quantitative real-time polymerase chain reaction, also known as qPCR, it comprises both amplification and fluorescent detection of amplified DNA target antimicrobial gene in the same step. Detection of fluorescence produced by a reported molecule or dye as intercalation increases, the detection of fluorescence is seen as the reaction proceeds. In this post-PCR gel electrophoresis is not required as in conventional PCR. And real-time monitoring progress of amplification is possible when process is ongoing, in contrast to conventional PCR method where analysis is possible after process is completed. I would like to just describe in brief the real-time PCR probe technologies which are available. There is the cyber green dye, which is sensitive but not specific because it binds non-specifically to double-stranded DNA, where the resulting DNA dye complex absorbs blue light at 497 nanometers and emits green light at 520 nanometers. It detects accumulation of any double-stranded DNA product. Among the fluorescent probe technology, there are the 5' nuclease Tachman probes, molecular beacons, dual FRET hybridization probes. Here, emission quenching relies on the interaction between electron excitation states of the two fluorescent dye molecules. So this is a diagrammatic representation of the probes. The one is the 5' nuclease probe in which there is a short oligonucleotide DNA. The 5' has the fluorescent dye and the 3' has the quencher. These bind to the complementary stand at 60 degrees centigrade. The Taq polymerase must cleave the 5' end of the Tachman probes, separating the fluorescent dye from the quenching dye, and then the cleaved dye accumulates after each PCR cycle. Then we have molecular beacons, in which they have a characteristic hairpin-like structure, in which the fluorescent dye and quencher dye are in approximation with each other. However, on encountering the complementary strand, they straighten out such that the fluorescent dye is then separated from the quencher and fluorescence is generated. This is a dual FRET hybridization probe in which two DNA probes anneal head to tail configuration. The upstream probe has a fluorescent dye on the 3' end, and the downstream probe has an acceptor dye on the 5' end. 3' end is phosphorylated to prevent its use as a primer. When both probes anneal, the fluorescence of the 3' end is absorbed by the acceptor dye on the 5', which then releases another wavelength. This slide shows how PCR may be used as a diagnostic tool for multi-drug resistance. This data is from Maureen M., published in Expert Review of Molecular Diagnostics, use of real-time PCR as a diagnostic tool for bacterial diseases. Here we can see that qPCR may be used as a diagnostic tool for methicillin-resistant Staph aureus, as well as for vancomycin-resistant Enterococcus. And these are the various target genes which are amplified, and the probe types used are as shown here. In addition, detection of genes encoding carbophenamases like BLA, KPC, VIM, OXA48, AIIMS, and metallobeta-lactamase can also be detected via qPCR. This is a study which was done by Zheng et al., published in Annals of Clinical Microbiology. In this, they have described the establishment of a duplex real-time PCR assay for rapid and simultaneous detection of NDM and KPC enzymes in bacteria. So in this, 157 Enterobacteriaceae isolates were collected. All isolates had previously been tested for BLA, KPC, AIIMS, VIM, NDM, and OXA48 using conventional PCR. 17 carbapenem-resistant isolates harbored the carbophenamase genes, KPC, AIIMS, and NDM. Then the Tachman probe-based real-time PCR assay was performed on ABI Prism 7500 fast apparatus. The assay initially validated using recombinant plasmids pNDM1 and pKPC2 carrying the NDM1 and KPC genes. Each run contained a positive control, which was a mixture of those recombinant plasmids, as well as one water blank as negative control. So the duplex PCR assay showed 100% concordance with the conventional PCR previously identified. So this shows us that real-time PCR required less time and labor for simultaneous detection of multiple carbophenamase genes, including NDM and KPC. Coming to multiplex PCR, this is a variant of PCR. In this, greater than one target sequence is amplified using multiple sets of primers within a single PCR mixture. This enables amplification of several gene segments at the same time. In this, primer lengths are within 18 to 25 nucleotides. Melting temperature of the primers need to be identical or within 1 to 2 degrees centigrade. And the GC content of the primer should be appropriate, around 50 to 55%. To avoid interference, primers should lack cross-complementarity. Repetitive sequences, regions with high homologies, and single nucleotide polymorphisms are to be avoided. So this is an old study which was done in the year 2003, and it describes a multiplex PCR assay for simultaneous detection of 9 clinically relevant antibiotic resistance genes of Staphylococcus aureus. And the results in this case were available within 6 hours. This includes the MEK-A, methicillin resistance, ACAPHD, aminoglycoside resistance, TET-K, TET-M, tetracycline resistance, A, C, macrolide lincosamide streptogrammin B resistant, VAT-A, VAT-B, and VAT-C, that is streptogrammin A resistance. Another feature is the development of this rapid multiplex PCR-based assay, which is the micro-B scan. This study was done by Liotti et al., and they developed a multiplex PCR platform for rapid detection of bacteria, antibiotic resistance, and candida in human blood samples. This was published in 2019. And in this, 31 out of the 39 culture documented bloodstream infection cases were concordant with this microB scan, because it was compared with the blood culture results. And it was found that changes made in antibiotic therapy for 20 patients could have taken place within an average of 9.7 hours in case of the microB scan assay result versus an average of 21.3 hours following blood culture, which makes it a desirable PCR, multiplex PCR platform. So this is an example of how whole blood sample can be processed, that is DNA extraction is done and multiplex qPCR is conducted, and within 5 hours one can get data with regard to antibiotic resistance and thus start the drug dosage accordingly. Relevance of PCR-based methods. qPCR and multiplex PCR are affordable, sensitive, specific, user-friendly, not space demanding, and deliverable. They deliver results in a few hours. They can be applied directly to clinical samples. However, these methods do not provide information about biochemical activity of enzyme involved in antibiotic resistance, and the phenotype associated with enzyme is not validated. Coming to DNA microarray technology, in this the principle involves again hybridization between nucleic acid strands. Oligonucleotide probes are designed to identify species and mutations conferring antibiotic resistance, which are then covalently linked to solid substrate in lattice pattern. The DNA sample is extracted from clinical isolates and labeled using fluorescent dyes. Complementary nucleic acid sequences between sample and probe attached on the chip is paired via hydrogen bonds, and then the fluorescently labeled target sequences bind to a probe sequence to generate a signal. So DNA microarray technology allows simultaneous detection of a large number of resistance genes, and it allows prediction of the isolates' resistances to multiple antibiotic classes. Uses of DNA microarray technology have been found as the checkpoint ESBL KPC DNA microarray has been used for identification and detection of ESBLs and Klebsiella pneumoniae carbophenamases. The sensitivities and specificities of checkpoint ESBL KPC DNA microarray is 100% as compared to PCR. The time taken is 7 to 8 hours compared to conventional susceptibility testing, which is 54 hours. Then the checkpoint ESBL KPC DNA microarray also is used to detect and identify KPC resistance in hospital-acquired pneumonia. In addition to this, there is another DNA microarray technology which has been developed, which is Check MDR CT102 DNA microarray. And this is again for detection of most prevalent carbophenamase genes, that is BLA, NDM, VIM, KPC, OXA48, AIIMS, and ESBL related genes, that is SHV, TM, CTXM, and this gives results in 2 hours. This is as per a study which was conducted by Nas et al. Then there is MVplex and StaffPlex DNA microarray technology, which combine qPCR and DNA microarray. So MVplex detects nuc, MEC-A, ORF-X, VAN-A, VAN-B, DDL, and TUF. That is MRSA screening in nasal swabs that directly from the nasal swab. Another StaffPlex detects again MEC-A, AAC, ERM-A, ERM-C, TET-M, and TET-X, and the Panton Valentine leucocidin. Coming to next generation sequencing, this is a breakthrough in the study of antimicrobial resistance. Genetic determinants of antimicrobial resistance are studied in a single microbial genome. DNA and RNA, which is then converted to cDNA, can be used as sequencing material. Next generation sequencing involving DNA helps to identify presence of antimicrobial genes, while next generation sequencing involving RNA helps to identify global gene expression, including expression of antimicrobial resistance genes. Then this also uses bioinformatics tools such as ResFinder, which allows the identification of acquired antimicrobial resistance genes. In addition to this, next generation sequencing helps in metagenomic analysis, uh, in which 16S rRNA gene sequencing on various specimens like blood, stool, and sputum allow in-depth study of members of bacterial community. Then in next generation sequencing, identification and characterization of resistance genes encoding the ESBL, that is CTXM, BLA-TEM, and BLA-SHV can be done. Quinolone resistance, that is mutations in GYR-A, PAR-C, or QNR elements can be done. And aminoglycoside resistance, that is aminoglycoside modifying enzymes and 16S rRNA methylases can be found out. Also, carbapenemases can be found out by this method. Relevance of next generation sequencing is that the bacterial genome can be assessed within several hours. Single molecules generating long reads have now been made, which is an advancement versus the small, shorter size draft molecules. Single cells next generation sequencing circumvents need for pure microbial culture, and it can be applied to study human pathogens surviving in the environment but unable to grow on artificial culture media. But there has been an improvement in sample preparation, which shortens the turnaround time from weeks to 4 to 5 days as compared to the previous next generation sequencing technology. And it also allows phylogenomic studies for genotyping, which is important for clinical epidemiology and public health. However, it is an expensive procedure which precludes its use as a primary diagnostic tool, and it is less sensitive than culture or PCR when applied directly to clinical samples. The next procedure is fluorescence in situ hybridization. In this, the principle is that a DNA probe 18 to 25 bases long, fluorescently labeled, single-stranded oligonucleotide probe hybridizes to RNA of target organism. Identification is at genus or species levels. Each kind of RNA can be hybridized, but our RNA is best suited as ribosomes are numerous, allowing for the maximum or boosting of fluorescent intensity. Fluorescent in situ hybridization is a cheap and convenient option for identification and resistance testing of bacterial pathogens. It's most commonly used to target 16S rRNA because this is genetically stable and its domain structure has conserved and variable regions, and it has a high copy number. Also, ribosomes are numerous in protein synthesizing cells. One variation of the probes used are the peptide nucleic acid probes, in which these are DNA analogs with uncharged polyamide backbones with sugar phosphate, which are stable to degradation, and they hybridize to complementary sequences with higher affinity. So, fluorescent in situ hybridization, in which peptide nucleic acid probes are used, is called peptide nucleic acid fluorescent in situ hybridization, which reduces non-specific probe attachment due to the electrically neutral backbone of oligonucleotides, and is recommended for routine diagnostics due to higher degree of standardization. Uses of fluorescent in situ hybridization. It can allow resistance testing from primary material, including tissues. It is particularly suitable if ribosomally mediated resistance is guaranteed. FISH is successfully applied if one or few variable bases are there, providing resistance. It is used for ribosomal mediated resistance to drugs like macrolide and linezolid. Clarithromycin resistance in H. pylori mediated by 3-point mutations in ribosomal 23S rRNA can be found out with fluorescence in situ hybridization. In this, 3 FISH probes are used, CLA R1, R2, and R3. R1 is associated with minimum inhibitory concentration of greater than 64 milligram per liter. R2 and R3 are associated with varying minimum inhibitory concentrations between 8 and 64 milligram per liter. So combination of combined use of these probes allows identification of co-infections with clarithromycin sensitive and clarithromycin resistant H. pylori by FISH. Sensitivity for detection of clarithromycin resistance is 90% and specificity is 100% by fluorescence in situ hybridization. In addition, fluorescence in situ hybridization has been used for clarithromycin resistance testing for thermotolerant Campylobacter species. In this wild-type probe and clarithromycin resistance probes targeting mutation in 23SR RNA, that is adenine to guanine at 2059, is used. In addition, linezolid resistance in enterococci, typically caused by the mutation 2567G to T base substitution in 23SR RNA can also be interpreted by means of fluorescent in situ hybridization. However, the relevance of fluorescence in situ hybridization is not very much in clinical practice because very few indications are there in which protocol for fish have been described. As I have mentioned above, standardization of fish-based resistance testing is also missing. Coming to phenotypic methods of non-culture based detection of antimicrobial resistance, we have the mass spectrometric approaches, which mainly includes the MALDI-TOF-MS, which stands for matrix assisted laser desorption ionization. In this, and TOF stands for time of flight. In this soft ionization method, samples are fixed in a crystalline matrix and are bombarded with an ultraviolet laser. The sample molecules vaporize into vacuum while being ionized at the same time without fragmenting or decomposing. So the time of flight is that it separates ions by their mass to charge ratio and determines that mass to charge ratio by the time it takes for the ions to reach a detector. And this generates characteristic mass spectral fingerprints, which is then compared with a large library of mass spectra. Spectral fingerprints form unique bioinformatics pattern profiling, which has to be referred to in order to understand the resistance, the molecular change in structure, which results in resistance. So this is a diagram showing the principle of MALDI-TOF-MS. Again, here we see that MALDI-TOF-MS is usually used as a culture-based technique, but it can be used as for processing direct clinical specimens, mainly from urine, CSF, and blood. But for other samples, they are generally done via culture techniques, not by non-culture techniques. So in this MALDI-TOF, detection of antimicrobial resistance basically is through three methods: detection of antibiotic modification due to enzymatic activity of bacteria, and also specific biomarkers which are discovered for antibiotic resistant strains, and subtyping strategy by MALDI-TOF-MS peptide protein profile of the cell. Detection of antimicrobial resistance mechanisms by measuring the enzymatic activity of bacteria. In this, accurate determination of structure of antibiotic and reaction metabolites has to be undertaken. The mass peak most likely present is that of the molecular ion peak that represents the exact mass of the antibiotic. Antibiotics and their degradation products are measured in the mass range of 100 to 1000 Dalton. In the ionization process, matrix usually adds proton to the antibiotic, but can also add sodium and potassium ions, increasing the mass of the antibiotic by 1, 23 or 39 Daltons respectively. Other reactions which take place in case of enzymes which denature the antibiotics include ionization of antibiotics, decarboxylation, decarbonylation, and loss of other groups of molecules. The mass peak patterns are unique to each antibiotic and can be used to detect antibiotic resistance. So this is a study which was conducted by Oviano et al., in which they have described rapid direct detection of carbapenemase producing Enterobacteriaceae in clinical urine samples by MALDI-TOF-MS analysis. Oviano et al. Developed an interpretative automated MALDI-TOF-MS based method for the direct detection of carbapenemase producing Enterobacteriaceae in clinical urine samples within 90 minutes of sample reception. And the assay yielded direct, reliable identification of 91%, that is 503 out of 553 samples, and showed 100% sensitivity and also 100% specificity for detecting carbapenemase activity in these samples. This shows a flow chart as to how much time was taken, the turnover time for this particular MALDI-TOF MS assay. In this, 3,041 urine samples were received, and then they were subjected to flow cytometry. Only those which had a count of greater than 1.5 into 10 to the power 5 bacteria per ml were then used. That is, in that case, 600 urine samples were then included in the study, and they were processed by MALDI-TOF MS as well as culture. So we find that the turnaround time for MALDI-TOF MS, it gave the direct ID of the bacteria as well as direct carbapenemase resistance detection was done, and it was done within 90 minutes, whereas the culture process took entirely 48 hours. So the invalid samples were those in which the amount of pellet obtained was not sufficient to carry out direct identification of bacteria and carbapenem resistance assay. That is, these are the invalid samples. Urine samples were considered positive with 10 to the power 4 CFU per ml of one microorganism. Samples with 2 or more microorganisms were not considered in the study, as they were considered contaminated. And these results which were obtained through MALDI-TOF were further again confirmed by PCR and further sequencing of carbapenemase genes. In MALDI-TOF antibiotic susceptibility testing, another very important part is the AMA modification. So in this ciprofloxacin, we find that ciprofloxacin or norfloxacin can be acetylated, and this causes inactivation and increase in its mass by 43 Dalton. And the acetylation is done by a particular enzyme, which is called aminoglycoside acetyl transferase type 1 BCR variant. So this is a plasmid-mediated quinolone resistance mechanism, which is most common in Enterobacteriaceae. The ratio between the sum of areas of the acetylated antibiotic and the intact form for ciprofloxacin and norfloxacin is seen, and thus the inactivation of this antibiotic is studied. It is an easy and fast technique and gives results within 30 minutes. The concept is the same as detecting antimicrobial resistance by measuring enzymatic activity of bacteria. However, interpretation of spectra are yet not fully developed. This was again a study conducted by Aviano et al. In addition, we can also use specific biomarker discovery for antibiotic resistant strains. This also has not been studied considerably. However, there is literature with regard to this, that is detection of MRSA, a small peptide called phenol-soluble modulin, which is encoded on type 2, type 3, and 8 SCC present in genomes of MRSA can be detected directly without culture-based techniques. The peptide is excreted by AGR-positive strains, and MALDI-TOF MS spectra shows a peak at 2415 Dalton. However, the sensitivity depends on the prevalence of this cassette in certain geographical areas. Then coming to the subtyping strategy by MALDI-TOF. In this, protein profile is used to detect phenotype-related biomarkers such as drug-resistant strains or similar species identification. Subtyping strategy uses the protein profile to obtain the subtype information using protein profile after strain identification has been done. So it can be applied to the identification of drug-resistant strains or the accurate identification of bacterial species complex. The relevance of MALDI-TOF is that it is high throughput and it's cost effective, and it has a high degree of accuracy. Reliability and accuracy of MALDI-TOF MS have been verified in a number of studies. Precision of microorganism identification is critically dependent, however, on the number of database entries, and therefore continuous updating of reference database is indispensable for advancement of this performance. Thank you.
So, Maitri, this is a question from Sarita ma'am.
Yes.
What do you mean by acquired resistance?
The acquired resistance is the bacteria developing resistance to a certain class of antibiotics to which it was earlier sensitive, and the mechanism can be through plasmids or transposons or mobile genetic elements.
And another question is, what is the difference between SNP and mutation? SNP and mutation. Single nucleotide polymorphism and mutation. We just add it in your seminar. Okay? Chalo. No any other questions? Okay.
Another question. Thank you.